Welcome to lesson 2.52, DNA replication. You guys have already told me that DNA, one of the functions of DNA, is to store information. So one function is to store information. But something that Watson and Crick noticed about the molecule as they built their model was that DNA, based off of Chargaff's rules, was going to be able to copy itself. And so DNA must be able to copy itself or its info in order to pass its information along to new cells. So before we divide a cell, we have to replicate the DNA or copy the DNA. So copying DNA is called DNA replication. And this process is known as being semi-conservative. And what that means is I'm going to conserve half of my molecule, half of my parent molecule, and the other half is going to be new, so that when I'm done, I have half old and half new. This side is conserved, this side is new. So every new DNA molecule is made up of half parent template and half new nucleotide string. All right? So semi-conservative means that half is old and half is new. We're conserving half of it. So when we build this new molecule, when we copy this molecule, we're doing it with several different enzymes. The first enzyme is helicase, and helicase unzips the DNA by breaking the hydrogen bonds. Once we break the hydrogen bonds and unzip the molecule, we have to hold the molecule apart, and we do that by something called single-stranded binding proteins, or SSBs. And single-stranded binding proteins are going to hold the DNA open. We've got to hold open that DNA molecule so that our other enzymes can come in and build the new strands of DNA. This fork that happens here is called the replication fork, and you kind of see a fork happening here. That is the replication fork, and we're going to have one on both ends of the DNA molecule. And we have to realize that, remember that we said that the DNA molecule that Watson and Crick built, they noticed that the sides of the molecule are built in opposite directions, that they're anti-parallel. So we represent that with 3' and 5' ends. DNA polymerase is our main builder. It builds new DNA. Builds new DNA. It is our main builder. It can only build 5' to 3', and it can only build off existing nucleotides. So DNA polymerase is going to come through. It's going to build the new strand, 5' to 3'. So we're good to go there. And so it's going to build our new strand of DNA. Now on the top, if we go in the same direction, we're going 3' to 5', and I said that we can only go 5' to 3'. So on the top here, we're going to have to build backwards in these fragments. These fragments are going to be called our lagging strand, because it's going to lag behind this other strand on the bottom. This lagging strand is made up of what we call Okazaki fragments. Okazaki fragments, after they're built, are going to have to be welded together, and they are welded together by an enzyme called ligase. This is our spot welder. it spot welds the Okazaki fragments together. It bonds them together. The bottom strand, the strand that we originally built, is called our leading strand because it's going to lead the process in the direction that our replication fork and our helicase is moving. So here's a glance at our replication fork and our replication bubble from a distance. I've put down the 5' and 3'. Our helicase is going to come in and open up this DNA molecule going in both directions, creating this replication bubble. Our DNA polymerase comes in, builds 5' to 3', builds our leading strand. On the other side, we are building our lagging strand, 5' to 3'. And as we move along, our replication fork is going to continue moving, and our bubble is going to get bigger and bigger. And so there's my leading strand up top getting larger and larger, my lagging strand lagging behind getting bigger and bigger. And then on the other side, eventually, that lagging strand is going to become a leading strand. And on the top, then I get a new lagging strand. Alright, so we're going to have leading and lagging. It just depends on where the replication fork begins. Alright, remember that I said that DNA polymerase can only build 5' to 3'. And the other thing I said was we can only build off existing nucleotides. Well, if I look here at the bottom, or even up here at the top, I don't have any existing nucleotides. I don't have any existing nucleotides here. So we're going to use a new enzyme called RNA primase. And RNA primase is going to lay down what we call an RNA primer. And this just allows DNA polymerase something to build off of. So DNA polymerase can come in and build off of those RNA nucleotides. We're going to do the same thing up the top. We're going to build some RNA primer, build some DNA. RNA primer, DNA. RNA primer, DNA. Ligase is going to come in and link those Okazaki fragments together. And then we're going to bring in, there's my ligase, then we're going to bring in DNA polymerase again. And DNA polymerase, along with being our builder, so it builds, now it's going to check our DNA that's been built, and it's going to remove or replace the RNA with DNA. Alright, so there's my DNA polymerase is going to move through and take away those RNA primers. On the bottom here, I got a little bit of an issue. I take that RNA primer away, this DNA polymerase can still only move from 5' to 3'. And it can only build off of existing nucleotide strands. So, what are we going to do? If I take away that RNA primer, I'm left without some DNA. Some of my DNA has gone missing. Well, lucky for us, at the end of our DNA strands, we have something called telomeres. Telomeres are repeating non-coding sequences. These repeating non-coding sequences are at the end of the chromosomes, so that our chromosomes, if we have some erosion, some chromosome erosion, we're not losing any pertinent information. Alright? And we have a large enough telomere for about 50 replications, which isn't a whole lot. So what we can do is we can replace some of those telomeres, and here are some repeating non-coding sequences, but I can use a new enzyme to build up our telomeres. So I can add bases to the end of my DNA. Telomerase can build 3' to 5'. The benefit to this is that it's extremely fast and extremely accurate. E. coli has about 5 million base pairs and can be copied within about an hour, and then divide into 2 identical daughter cells. The human cell has about 6 billion bases before it can divide into new daughter cells, and that happens in just a few hours. So it's remarkably fast. It's remarkably accurate. We have only about 1 error per 100 million bases, or 30 errors per cell cycle, and we'll find out that some of those errors won't even matter. What we're looking at here on this picture is what it actually looks like within a cell, and you can see the replication bubbles here. This line is the DNA, and you can see the replication bubbles forming here and down here. And on this side, I just have a picture of those 4 replication bubbles that we can see. So study this. It's a lot of enzymes. Be able to tell the story from beginning to end. The helicase, the single-stranded binding proteins, the RNA primer, or RNA primase, DNA polymerase, building our leading and lagging strands, the Okazaki fragments being welded together by ligase, and then DNA polymerase coming back through, double-checking everything, and replacing those RNA primers with DNA. And then the telomerase coming in at the end and building up those telomeres. All right, be able to tell that story from beginning to end, and you guys will be fine. I'll see everybody in class.